Hello, biology students. We're now ready to continue our talk on RNA. We're going to be going to our next section, which is going to be looking at ribosomes and protein synthesis. And so, if you remember from last time, we talked about the fact that RNA is related to DNA, but there are some key differences. The first process is when we take the DNA to RNA, and so as you recall, that will be known as transcription. And so now we need to take the RNA that we made during transcription, and now we need to make it into protein, and so that it will be the process that's known as translation. And so what we're going to be looking at today, we're going to be looking at what the genetic code is and how it is read. We'll also look at the role of ribosomes and how they play in assembling proteins. And then we'll also talk about the central dogma of molecular biology and what that means to us. All right. We're going to look first at translation as the process. And very similar to transcription, there are going to be a few different steps. And the first part is initiation. And so we have the coming together of the ribosome complex and the tRNA. And so remember that tRNA stands for transfer RNA. And so each of the transfer RNA is going to have a specific amino acid associated with it. And that specific amino acid will then, depending upon how it's added, will determine the sequence of bases that is needed in DNA. Once we have that, we're going to have what's called elongation. And so the tRNA will then bind to the start codon on mRNA. And so the start codon is always AUG. One of my friends in college told me a good way to remember this is a unique go. And so if you want to use that as well, you can. And so AUG codes for the amino acid called methionine. And so every protein molecule starts off with the amino acid methionine. How does this work? And so what we have on the RNA, we have 3 letters, which is referred to as the codon, and then the tRNA, which is up here, is going to be base pairing with that to form what's referred to as the anticodon. So we have the codon of the RNA, the anticodon of the tRNA, through the base pairing. So if you remember, A goes with T, C goes with G, or in the case of RNA, A goes with U and C always goes with G. We will then have the binding of those 2 substances. It'll keep getting read along down the chain of the mRNA and will eventually stop or terminate once it reaches what's referred to as a stop codon. And so a stop codon, there are 3 different stop codons which basically do not code for any specific amino acid. And so once the termination has begun, then the ribosome complex will fall off, and then it'll then start the process over again. The amino acid chain that's formed is called a polypeptide, and so the polypeptide will then be able to fold to make the protein. I'm going to talk a little bit about the importance of the genetic code. And so the genetic code are the rules that will tell us what type of amino acid goes with the specific type of codon. And so I want to show you some examples. And so this is going to regulate how a sequence of nitrogenous bases in mRNA correspond to a specific amino acid. So for example, if we have the sequence AUG, which is what we always start with the unique go, we can use this table to figure out what amino acid this codes for. On the left side, we have the first position, the middle here is the second position, and on the right here is the third position. And so if we use AUG, we simply look for A, we have U, and then we have the G over here. And so if we come down here, we will see that AUG codes for methionine. And so we can do this with any other codon that we're interested in. Let's say, for instance, we're looking at CAU. So we have our first position is C, our second position is A, our third position is U. And so we can see that CAU codes for the amino acid called histidine. And notice that this will go along every 3 nitrogenous bases. So depending upon whatever the sequence of mRNA that was transcribed, We're going to then read them 3 at a time. And so every 3 amino acids, every 3 codons will be then used to make an amino acid. And so that is how the genetic code works. I do want to point out something very unique, though. The genetic code, regardless if you're a bacterium or if you're a human, is nearly universal. And so what this means then, AUG will code for methionine whether you're a bacterium or whether you're a human. CAU will code for histidine whether you're a bacteria or a human or any other organism. And so it does work the same in every organism. And so I really feel that this is, again, very strong proof of a higher intelligence than we are that has made this all possible. So go ahead and just kind of reflect on that for just a second to see how all of this can be used by any living organism. And so just think about that just for a second. All right, let us continue on. We have a picture here which shows what translation looks like. So we have the 2 ribosome parts, which are the green and the red. This part right here, this tape-looking thing, is your messenger RNA. Notice we have the transfer RNA. This is the anticodon region. These would be the codon regions of the RNA. Notice that these will slide into place, and basically the ribosome reads it along. At the ends of each of the tRNA, these balls are representing the different types of amino acids. And if you notice, you'll see that this sequence will continue to keep growing as it goes along. And so after it reaches the stop codon somewhere further down here, the ribosome, this part right here, it'll break apart, and then the protein will be released, and then it'll fold into its proper shape. And so this is the process known as translation. So taking messenger RNA and using the transfer RNA to then make it into a protein. Couple facts about translation. mRNA can be recycled and reused by another ribosome to create another protein, or it can be degraded. And so this is very useful for the body and the cell specifically, because therefore you're not using up any more energy than you need to. So the body is always looking for ways to conserve energy and therefore not having to burn through as much ATP, for example. And so the mRNA can be used many, many times over. There are also 20 common amino acids, and so fortunately for you guys, you will not have to memorize those, nor will you have to know the shapes of them. And so, as I pointed out before, the start codon AUG, a unique GO, always codes for the amino acid methionine. I want to now talk a little bit about what is referred to as the central dogma of molecular biology. And so this was first proposed by Francis Crick of Watson and Crick fame. And so what he noted was that you always start with DNA. The DNA is transcribed into RNA, and then the RNA is translated into protein. And so notice that for transcription, the DNA acts as a template for the synthesis of mRNA, and therefore it creates a message. Because we know the DNA is within the nucleus of a cell, therefore the process of transcription also has to be within the nucleus. Once the mRNA is made, it's transported out of the nucleus and ends up in the general cytoplasm. And so the second part of this process, which is translation, the mRNA directs the assembly of a protein, and therefore this part occurs within the cytoplasm. And so that is the differences then between those two. Right. Finally, I just want to talk about a couple things about the roles of RNA and DNA. DNA is considered the master plan. And so obviously, because it's important and every other cell contains this important information, you do not want to destroy or endanger it. And so it is important that it does in fact stay safely inside of the nucleus. RNA, however, is the disposable blueprint. And so as long as you have your DNA, you could always go back to that and use it then to make more RNA. And so in a sense, the RNA can risk being taken out into the field, which in this case is the cytoplasm of the cell, and so it can be exported out into the cytoplasm. And so again, in order to get protein, there does have to be the process of translation, and so that will take the mRNA and convert it one amino acid at a time to form a protein. And so if there's any questions, I'll be happy to answer them. And so this is Dr. S signing off.